and I'm a PhD student at the Department of Pharmacology and Therapeutics at the University of Manitoba. And today in this lecture, I will talk about Alzheimer's disease. And I've titled this presentation, Alzheimer's, a disease you won't forget, because people who suffer from Alzheimer's, they tend to forget things. So before I proceed, I would like to introduce myself and talk a bit about my background and my journey to Canada. So I was born in India, and here on top right, that's me. And then below that is me again working in the lab. So I have a bachelor's with honors in zoology from University of Delhi that I finished in 2017. And after that, I finished my master's from the same university in zoology in 2019. And when I was in my bachelor's and master's, I did 2 internships in science labs, which really helped me to get a PhD position. And the first internship was at a molecular oncology lab in 2016. And the second internship was at Department of Molecular Reproduction, Development and Genetics at Indian Institute of Science in Bangalore in 2018. And during these internships, I learned the process of drug development, how to culture a bacterial colony, how to isolate and clone DNA, and some other basic molecular techniques. And after my master's, I applied for a PhD here at the University of Manitoba in Canada and flew more than 11,000 kilometers across the globe to come to Canada. And I started my PhD in January of 2020 in the Department of Pharmacology and Therapeutics, University of Manitoba. And currently I'm working under the supervision of my advisors, Dr. Elbenzi and Dr. Fernie Yu, both of whose labs are at the Division of Neurodegenerative Disorders at St. Boniface Research Center here in Winnipeg. Since I want this lecture to be interactive, we will have polls like this every couple of slides. So the first question I have for you is, how are you feeling today? So you can go to slido.com and enter this code, or you could scan this QR code and then indicate how you're feeling today. Okay, we're getting good responses. So far, we have 23 people who've answered. We're just going to wait for a couple of more minutes, like a minute or so. All right, okay, so looks like the average score we are getting is 3.5, which is towards the positive side. And 44% of you are feeling happy. Okay, now 42%. Well, thank you for your feedback. And okay, we have more people answering. Oh, the overall score is going down now. Okay, we'll go back to the lecture. Okay, thank you so much for answering. All right, so the final score looks to be 3.3. That's still more than 2.5, so okay. Okay, now I want to have an idea of how many of you are actually from a biology background. So if you are currently in health sciences program or biological sciences, please answer yes or else no. Looks like majority of you are from a biology background. Okay. So far, we have 22 people who answered, and 86% of them are from a biology background. That's nice. I'm just going to wait like 30 more seconds, and then we'll start the lecture. All right, it looks like majority of you are from a biology background, but we still have some people who are not. Okay, thank you so much for answering to this poll. And I'll switch back to our slides now. Okay, so moving forward, here is the outline of today's lecture. So I'll start with the introduction, followed by signs and symptoms of Alzheimer's disease. And then I'll talk about risk factors of Alzheimer's disease, followed by stages and pathology, diagnosis, and treatment of Alzheimer's disease. And towards the end, I'll briefly talk about my research and what I do in the lab. And at the end, we will have a short online quiz, which is going to be very easy and will cover the main takeaways from this lecture. So we'll go through the entire lecture first and then summarize it with key points at the end, and then have a quiz from those key points. It will be a very basic quiz with questions like what happens in case of Alzheimer's disease. So let's get started. Our first topic is introduction. So what is Alzheimer's disease? Alzheimer's disease is a brain disorder that occurs in older individuals and begins with mild memory loss that can possibly lead to major problems with memory, thinking, and behavior that disrupts the daily life of an individual. So people suffer from memory loss in Alzheimer's disease. They have difficulty in remembering things. They start forgetting their close family members or friends or their own parents and children as well. One thing to note here is that it is not a normal part of aging. And what do I mean by that is that when people get older, natural changes do occur, which might include forgetfulness. But in case of Alzheimer's disease, this forgetfulness becomes much, much significant. For example, with aging, people might be unable to remember details of a conversation or an event that took place 1 year ago. But in case of Alzheimer's disease, they are unable to remember or recall details of events that happened like yesterday. Also, Alzheimer's disease is the most common form of dementia. And what dementia is that? Dementia itself is not any specific disease, but it is an umbrella term for diseases that affect memory and cognition. So we have frontotemporal dementia, Lewy body dementia, vascular dementia, and Alzheimer's disease. So all these diseases are diseases that lead to memory loss or problems with memory. But out of all these diseases, Alzheimer's disease is the most common form of dementia. Also, Alzheimer's disease is a progressive neurodegenerative disorder that occurs when neurons in the brain die. So our brain is composed of cells called as neurons. Here you can see a healthy neuron, which has a good outgrowth, and it helps the brain to communicate and also contributes to formation of memories. And with the progression of Alzheimer's disease, these brain cells called as neurons, they start to degenerate and eventually die, which is why Alzheimer's is called as a progressive neurodegenerative disorder, because neurons die in it or neurons degenerate. And it is really important to address this issue. And why do I say that is because of the increasing number of cases. Currently, there are more than 55 million people who live with such memory impairments worldwide. And just to give you a comparison, The total population of Canada is 38 million. And unfortunately, this disease not only affects the life of the individual suffering from it, but it also puts a lot of mental and physical stress on their families. One critical concern for those living with Alzheimer's is that they have a tendency to wander into unfamiliar surroundings and get lost. In fact, if we look at this graph here in dark blue, we can see the total number of people suffering from dementia and memory-related problems in Canada alone in 2020 in different age groups. And if we look at this one specific age group, that's 85 to 89 years, in 2020, we had around 150,000 people who were suffering from such memory impairments. And by 2050, this number is expected to go beyond 500,000. Hence, it is really, really important not to forget about this disease, in which people forget. This brings us to our first question. So please go to slider.com and answer, which is the most common form of dementia? Amazing. So these are the kind of questions you can expect towards the end of the lecture as well. Really easy, and you'll remember it as well. Okay, someone said vascular dementia. No, Alzheimer's disease is the most common form of dementia. So 97% of you have gotten it right, which is a really nice figure. Okay. Well, thank you so much for answering. And this is one takeaway from today's lecture, which is the most common form of dementia. Now we'll switch back to the lecture. Our second topic for today is signs and symptoms of Alzheimer's disease. So here are some signs of Alzheimer's. The first and the most important one is memory loss that can disrupt the daily life of an individual. For example, people tend to forget their own names and who they are, which is very concerning. Second sign is losing track of dates, time, and current location, and hence people tend to get lost as well. Another sign is problems with language and writing. The fourth one is a painful and heartbreaking one because people forget their close ones. So a person who has Alzheimer's, they'll start forgetting their own children, their siblings, even their spouse. And another sign is repeating same questions again and again. And for example, I was sitting with one of my colleagues and having lunch one day, and her father called her 5 times in an hour. So out of concern, I asked her, is everything okay? And that's when she told me that her father has Alzheimer's disease, and he's been calling her again and again to ask her how she's doing. And every time he calls her and hangs up, a few moments later, he forgets that he's already called her and calls her again. So people with dementia or Alzheimer's disease, they repeat same questions, and this is one sign of Alzheimer's disease. Poor judgment and misplacing things, like keeping things into unusual places, is another sign. And lastly, there are changes in mood, behavior, and personality. For example, say if you met a person with Alzheimer's disease on day 1, and when you meet them again the next day, you'll feel like they are not the same person you met yesterday, because personality change happens in Alzheimer's disease. Our third topic is risk factors for Alzheimer's disease. Risk factor means that's something that increases the chance of developing a disease. And the most important risk factor for Alzheimer's disease is increasing age. Hence, older people are at a higher risk of Alzheimer's disease. Second one is environmental factors or lifestyle factors, like cigarette smoking increases the risk of having Alzheimer's, or the kind of environment you're working in. So if you're working in an environment wherein you're getting exposed to toxins or viruses, it also increases the risk of acquiring Alzheimer's later in the life. Third one is gender. It is important to note that older females are at a higher risk of Alzheimer's disease than older males. And this is in part because older females go through menopause. And with menopause, there's a decline in a very important hormone called estrogen in their body. Hence, older females are at a higher risk of Alzheimer's than older males. In fact, 2/3 of the people who are suffering from Alzheimer's right now are females. So gender is another risk factor. Next one is genetics and heredity. So our genes are made up of sequences of DNA, and that is what a parent passes on to their children. And there is this gene called as ApoE gene that provides instructions for making a protein called as apolipoprotein E, which is responsible for carrying fats and cholesterol throughout the bloodstream. And this ApoE gene exists in 3 isoforms in humans: ApoE2, ApoE3, and ApoE4. And the presence of ApoE4 is a genetic risk factor for development of Alzheimer's disease and has been found to be expressed in more than half of the people suffering from Alzheimer's. So let's say if someone has ApoE4, it doesn't really mean that they will get Alzheimer's disease, but what it means is that they are at a higher risk of getting Alzheimer's disease as compared to people who don't have this ApoE4 isoform. And the next risk factor is traumatic brain injury, which occurs in car accidents or when people fall down from heights. That leads to concussion or clots in the head, and eventually the cells of the brain die. So traumatic brain injury also puts you at a risk of Alzheimer's disease. Some other risk factors include diabetes, obesity, poor diet, lack of physical activity, and high alcohol consumption. So these were the risk factors of Alzheimer's disease, with aging being the most important risk factor. Now moving on to stages of Alzheimer's disease. Alzheimer's disease is broadly classified into 3 different stages based on the progression of the disease. The first one is early stage Alzheimer's, where the symptoms are mild. So this stage is generally asymptomatic, and minor difficulties occur in this stage. And they include like difficulty in remembering the right word or right name of someone. They'll have difficulty in performing everyday tasks with ease, or they'll forget the location of everyday objects. And they'll also have difficulty in remembering names when they're introduced to new people. But these symptoms can only be noticed by close family or friends like who are living with them, and they are not very prominent. So the symptoms are mild in early stage Alzheimer's. Moving on to mild stage Alzheimer's, the symptoms become moderate. And this is the longest stage that can last for years. This is the stage where symptoms get more pronounced and nerve cell damage starts. So people will start forgetting their personal history, or they'll forget their cell phone number that they've had for like 10 years, or they'll also forget their own house address in which they have lived for, say, 20 years. They start feeling confusion about day or date, and they also experience changes in sleep pattern. So these are the symptoms of middle stage Alzheimer's. And moving on to the late stage Alzheimer's, that is severe, the symptoms become much more pronounced. They start having difficulty in communicating and also lose control over body movement. Memory and cognition, they decline significantly, and personality changes can also occur. In this stage, patients need extensive care. Some of the symptoms include difficulty in having even basic communication. They start losing the awareness of their own surroundings. That is why they get lost. They start forgetting their close ones, like their children or their spouse, their family members with whom they have lived for like 50 years. Other symptoms include weight loss, and seizures can also happen. So these are the 3 stages of Alzheimer's: early stage, middle stage, and late stage, or mild, moderate, and severe, with symptoms getting bad from mild to moderate and worse from moderate to severe stage. Now I have a question for you. And the question is, what is the biggest risk factor for Alzheimer's disease? So let's go to slido.com and start answering that question. Great, everyone's answering correct, and that's really nice. Okay, so 33 of you have answered, and everyone's answered the right one. That's increasing age. So congratulations. And now we'll move on to the lecture. Okay, 35 people answered. Great, thank you. Now our 5th topic for today, that's pathology of Alzheimer's disease. Pathology is the science of the cause of a disease. So now we'll see what causes Alzheimer's disease. So there are 2 major pathological hallmarks of Alzheimer's. This means that these 2 things are what mainly cause Alzheimer's. The first one being NFTs, that stands for neurofibrillary tangles, and the second one are amyloid beta plaques. So first I will talk about NFTs, neurofibrillary tangles. So these NFTs are intracellular. That is, they get formed inside the cell and are made up of insoluble, twisted fibers composed of tau protein. So here, in healthy neurons, tau that you can see in blue, this tau normally binds and stabilizes the microtubule that provides structure to the neuron. But in case of Alzheimer's disease, due to abnormal processing of this tau detaches from the microtubule and sticks to other tau molecules, forming neurofibrillary tangles inside the neurons. This causes the microtubule structure to collapse, which ultimately kills the cell and contributes to death of neurons. And now we'll quickly see how this tau protein becomes abnormally twisted and tangled. Here in blue, we can see the enlarged neuronal microtubule structure that is stabilized by normal tau protein in red. But due to abnormal processing or the abnormal action of certain enzymes, it causes hyperphosphorylation of tau, which means increased addition of phosphate group to these tau proteins. And hence tau detaches from these microtubules and sticks to other tau molecules and eventually forms neurofibrillary tangles. This leads to distortion of cell structure, and now the neuron cannot transport nutrients and other important substances from one end to another. Hence, the microtubule structure collapses, leading to death of neurons. Hence, from a healthy neuron, we can see how it becomes a degenerated neuron, and neurodegeneration occurs in Alzheimer's. Now moving on to amyloid beta plaques. So these are extracellular aggregates of misfolded amyloid beta proteins that accumulate between the neurons and trigger an immune response that leads to inflammation and causes cell death. These amyloid beta proteins are formed due to the abnormal processing of a protein called as amyloid precursor protein. Also, testing for the presence of increased levels of amyloid beta plaques in the brain is an important part of Alzheimer's diagnosis and research. Now let's see how these amyloid beta plaques are formed. So there is this protein called as APP that stands for amyloid precursor protein present on the cell membrane. And this APP is being acted upon by alpha secretase, followed by gamma secretase, which leads to the generation of a P3 fragment, which is non-toxic. And this pathway of amyloid precursor protein processing is called as non-amyloidogenic pathway. And this leads to production of a non-toxic P3 fragment. Whereas moving on to the other amyloidogenic pathway, what happens is that this amyloid precursor protein is acted upon by beta secretase instead of alpha secretase. So APP, when acted upon by beta secretase, followed by gamma secretase, leads to production of amyloid beta fragment that clump together and forms amyloid beta plaques that collect between neurons and disrupt cell function, eventually leading to cell death. So to summarize, what we just saw was that intracellular neurofibrillary tangles accumulate inside the cell and disrupt microtubule structure that leads to impaired transport system within a neuron, while extracellular amyloid beta plaques deposit between the neurons and disrupt neuron-to-neuron signaling. Hence, both of these hallmarks, they contribute to cell death, leading to neurodegeneration and contribute to progression of Alzheimer's disease. One mechanism by which amyloid beta plaques lead to neurodegeneration is through neuroinflammation. So the accumulation of these amyloid beta plaques in the brain leads to the activation of immune cells called as microglia. While these microglia try to clear these amyloid beta plaques from the brain, at the same time, they also release inflammatory cytokines, or you could say inflammatory signals, so that more immune cells can come to rescue. But because there is too much amyloid beta, these cells are unable to clear all the amyloid beta, and this leads to a sustained inflammation, which is also called as neuroinflammation because it's happening in the neurons. And this neuroinflammation drives neurodegeneration. So along with amyloid beta plaques, neurofibrillary tangles, we also see neuroinflammation in case of Alzheimer's disease. And as a consequence of all this, we see neurodegeneration. Here on the left is a normal brain, and on the right is what happens to the brain during Alzheimer's disease. So due to death of brain cells and neurodegeneration, the brain regions start to shrink. So we can see the cortex shrinks, whereas the hippocampus, the structure right here that stores memory, that also shrinks in case of Alzheimer's disease. Whereas these ventricles, which are large fluid-filled spaces in the brain, they also start to shrink. They become bigger, in fact. So these fluid-filled spaces become bigger, whereas the tissue starts to shrink in Alzheimer's disease due to neurodegeneration. And here we have an animation showing how the brain shrinks in Alzheimer's disease. So now there's another question for you. What are the hallmarks of Alzheimer's disease? I also see someone has asked a question in the chat. So I'll go through all the questions towards the end of the lecture. So if you have any questions, just feel free to type it in the chat and we'll go through them at the end. And looks like everyone's gotten this right. So yes, the major hallmarks of Alzheimer's disease are both neurofibrillary tangles and amyloid beta plaques. So you guys are doing amazing. Okay, moving back to the presentation. Now our sixth topic for today is diagnosis of Alzheimer's disease. And there are multiple methods and tests designed to diagnose Alzheimer's disease. The first and the most common one is memory and problem-solving tests. These tests evaluate memory, thinking, and simple problem-solving abilities to assess the changes in behaviors and cognition. Most of these tests give an overall sense of whether a person is experiencing cognitive symptoms or not. Some examples of these memory and problem-solving tests include MOCA, which stands for Montreal Cognitive Assessment, and MMSE, that stands for Mini Mental State Exam. The next way to diagnose Alzheimer's is through blood tests. So blood of the patient who complains about memory loss or related symptoms is collected, and the levels of certain biomarkers like amyloid beta is measured. There are few blood tests that are currently available on the market that can be ordered by healthcare providers to aid in the diagnosis of memory complaints. And a blood test can also be used to look for the presence of ApoE4 gene, which is the risk factor for Alzheimer's disease. And lastly, brain scans like magnetic resonance imaging or PET scan and even CT scan can also aid in diagnosis of Alzheimer's disease. These tests also help in ruling out other conditions that may cause symptoms similar to Alzheimer's. And these tests can reveal tumors or the evidence of stroke or head trauma. So all these three diagnostic tools help to diagnose Alzheimer's disease. Now, moving on to our final topic for today, which is treatment of Alzheimer's disease. There are several drugs out there which aim at slowing the progression of Alzheimer's. And today, we'll learn about only three of them or three categories of them. So first one is donepezil, which is an inhibitor of enzyme called as acetylcholinesterase. So what it does is that two neurons communicate via tiny molecules called as neurotransmitters. And the enzyme acetylcholinesterase breaks these neurotransmitters. What donepezil does is that it inhibits the action of this enzyme, and hence, as a result, it enhances the neurotransmitter transmission between two neurons, which relieves the symptoms of Alzheimer's disease because we have more neurotransmitters between two neurons now, because this enzyme has been inhibited by this molecule or this compound. Another clinically useful drug is memantine. It is effective in many neurological disorders, including Alzheimer's disease. And the principal mechanism of action of memantine is that it prevents too much calcium from moving inside the brain cells. High amounts of calcium in cells can lead to cell death, and hence memantine prevents that by blocking certain receptors that allow calcium to flow inside the cell. And finally, I would like to talk about lecanemab. Well, this drug recently received FDA approval on January 6, 2023, which is 12 days ago. And what this drug does is that it selectively binds and eliminates one of the hallmarks of Alzheimer's disease, that is the toxic amyloid beta plaques. Hence, clearing these amyloid beta plaques from the brain reduces inflammation, improves neuron-to-neuron communication, and slows down the progression of Alzheimer's disease. And that was all from Alzheimer's disease for today. And now in the next 4 slides, I will briefly talk about research to give you an idea about how it is done. So first and most important thing when you plan to conduct research is to have two groups. That's control and experimental group. And what are these groups? Well, an experimental group, also known as the treatment group, is the one that receives the drug treatment whose effect you want to study, whereas the control group does not get any treatment. For example, if I want to study the effect of high-fat diet on obesity, I will have one control group and one experimental group. So what will happen is that the control group will eat only a normal diet, like what they were using or what they were eating previously. But the experimental group will be fed a high-fat diet. And after a certain period of time, results will be analyzed to see how the intake of high-fat diet affected the progression of obesity in experimental group versus the control group. That's why these two groups are really important in research. And now I'll quickly tell you about my research. So my research is preclinical. And what preclinical means is that we do not work with humans, but rather we work with animals. So preclinical, pre standing for before, and clinical means clinics. So since it's not in the clinics, we cannot work with humans, but we work with animal models. And I work on Alzheimer's disease. So to study Alzheimer's disease, I use mouse models of Alzheimer's disease. These mice are genetically modified. That means that their DNA has been altered in such a way that they possess symptoms of Alzheimer's disease like memory impairment, neuroinflammation, and amyloid beta pathology. And what I do next is that I surgically take their brains out and isolate the brain cells called neurons. Then I grow these cells in cell culture plates, followed by my treatment and evaluation. For example, I perform treatment with the drug estrogen. And since inflammation contributes to Alzheimer's disease, I'm hoping that my treatment will decrease inflammation in these brain cells and hence improve the symptoms of Alzheimer's disease. So the next question that arises is, after I give my treatment, how do I evaluate inflammation? So let's talk about that. And before we go ahead, here is an image that I captured using a confocal microscope. And these are some neurons that I obtained from the brain of a mouse. So here in blue, you can see the nucleus, and this is the cell body of the neuron. And these are all the axons and dendrites, or you can say neuronal processes. So that's how a neuron looks under the microscope. So in the previous slide, I was talking about how to evaluate inflammation after treatment. So here we have the NF-kappa B pathway that is considered to be pro-inflammatory, meaning that the activation of this pathway in cells is going to lead to inflammation. So as you can see in this image, we're not going to talk about this part, but we'll just focus on here to keep it simple. So as you can see, upon activation, these subunits called P65, P50, and P52 and REL-B. So these NF-kappa B subunits, they move inside the nucleus and their levels increase here. Then they affect gene transcription, which eventually leads to inflammation. So because what I am hoping is that my drug treatment will decrease inflammation, I would be interested in studying or looking at these specific subunits inside the nucleus. One thing to note here is that we have tools and techniques using which we can measure the amount of these individual subunits. So I have molecular techniques using which I can measure P65 or P50 or both of them. So what I'll do after my treatment is that I will collect the entire protein from the cells, and by using molecular techniques, I will measure the levels of these NF-kappa B subunits inside the nucleus. And if the levels of these subunits decreases after my treatment, I can say, yes, the inflammation is going down. Or if the levels of these subunits is increasing, I'll be like, oh no, my treatment is promoting inflammation, and that's something I don't want. And yeah, just like we have NF-kappa B pathway to study inflammation, there are several other pathways that we can study, that we can use to study different cellular aspects like energy, growth, and much more. So just by looking at some of the subunits in a specific pathway, we can have an idea of what's going on inside the cell or how our treatment is working. And finally, let's talk about clinical trials. So unlike preclinical studies, clinical studies or clinical trials use human beings or human subjects. But before clinical trials, preclinical studies are conducted, and only after preclinical studies give you promising results, things proceed into clinical trials. So once the patients sign up for a clinical trial, they are assigned into 3 groups. So we just learned about control and treatment group, but here we have another group called as placebo. So placebo is a group that receives what to them appears to be the treatment, but is actually not. And this is what allows the researchers to compare the results of the treatment and make sure that the changes that they are seeing in the treatment group has resulted from drug treatment and not just by chance. I'll explain this further. So after patients are assigned into these groups, they receive the treatment and follow-up is done, and then the results are compared. So for example, if you see changes after treatment in both the treatment group and the placebo group, this will mean that the changes are not because of the drug, because placebo never received the drug, and hence further testing of the drug will be required. But if you see changes only in the treatment group and nothing in placebo and the control group, that's when you can say that, oh yes, my drug is working, because the changes are only in the treatment group that received the drug. And hence we can proceed further with that. Drug, and further testing can be done on like a larger population. With this, we can go over the main key points of today's lecture. So the first one is Alzheimer's disease is a neurodegenerative disorder. What it means is that in Alzheimer's, neurons in the brain degenerate. That's why it's called a neurodegenerative disorder. Also, Alzheimer's is the most common form of dementia. So we have different diseases that lead to memory impairments. Out of all those diseases, Alzheimer's is the most common one. Next one is that people suffer from severe memory loss in Alzheimer's disease. So this is what happens in Alzheimer's. People lose memory and they start forgetting things. After that, age is the biggest risk factor for Alzheimer's. So people who are getting older are at a higher risk of Alzheimer's than younger people. And in that too, older women are at a higher risk of Alzheimer's than older men, because women undergo menopause when they get older, and then they face a decline in the levels of hormone called as estrogen, which is important for memory formation, which is why older women are at a higher risk than older men. There are 3 stages of Alzheimer's disease: mild, moderate, and severe, with symptoms getting bad from mild to moderate and worse from moderate to severe stage. We studied about 2 major hallmarks of Alzheimer's disease, the neurofibrillary tangles and the amyloid beta plaques. And we also studied that Alzheimer's can be diagnosed using memory tests, blood tests, and brain scans like PET, MRI, or CT scan. Also, the latest Alzheimer's drug is lecanemab that targets the toxic amyloid beta plaques, which is one of the hallmarks of Alzheimer's disease. And finally, preclinical studies do not involve humans, while clinical studies do. So I do preclinical studies, and I study with animal models, whereas clinical trials use human subjects. The next slide, which is the thank you slide. So this brings us to the end of our lecture today. I would like to thank all of you for your time, and I hope you are leaving with a better understanding of Alzheimer's disease today. And if you have any questions after this lecture, we will stay and chat about any questions if you have. or if you want to email me, you can reach out to me on mishrap@myumanitoba.ca, and I will try to get back to you as soon as possible. And now we seem to have about 5 minutes left, so I'll be happy to take any questions.